Well, we have finally made it to the end part of Chapter 4. This is our third installment about protein structures and functions. And now that we have become somewhat familiar with how proteins go about obtaining their structure and how exactly they function through the interaction with other molecules within the body, let's go ahead and take a look at exactly how we can control when proteins are turned on and off within our cells. And also towards the end, we'll talk a little bit, not too much, about exactly how we go about studying proteins. Now, I hope by now you realize that a lot of our chemical reactions within the cell are reliant on a cascading effect, meaning that it will often rely on multiple enzymes or multiple proteins from start to finish to get to your final product. So one of the ways that we can control when proteins are turned on and off is to what we like to call a feedback system. So oftentimes we see that the product that's produced can be used to relay back to the enzymes and the protein utilized in the sequence to shut them down so that they don't continue functioning and produce any excess product. Now, a feedback system basically relies on a stimulus and a product, and it can go two ways. We can have what we call a negative feedback system, which is what our body prefers, and you can have a positive feedback system. Let's go ahead and take a look at some of these illustrations so that I can better explain to you what a feedback inhibition is all about. So here on our left-hand side, we're going to go ahead and we're going to take a look at a reaction that is going to require you to produce your A, B, and C products. And what we see happening is that it's a cascading or a step-by-step reaction because it's going to involve several players. Now, when we get to the production of B, we see that once B is produced, it will go ahead and send off a signal that will cause the activation of X, Y, and Z. Now, Z is our final product. You can see that's the last one that's being made into the chain, right? And one of the ways that we can go ahead and control within the cell exactly how much Z product we produce is through a feedback inhibition. And what we see happening here is as soon as the reaction produces the product that we're after, in this case Z, the product's presence will go ahead and send off a signal that will inhibit the chain from occurring anymore. So in this case, it will send off a signal that will shut down X. And if X shuts down, then Y shuts down. And as an end result, we don't produce any more Z. So you basically only work until you get the product. And once you have your product, we see that that product will inhibit the first enzymes in the step and allowing them to shut down. So thereby we can limit how much concentration of the product is found within the cell. This is a very energy favorable type model because the cell, or I should say the mechanics, are only active long enough to produce the product that is needed. It is also quite easy to reverse the reaction because oftentimes what we see is as the quantity or the concentration of the product starts dwindling down, the inhibition signal is taken away, and then the mechanisms are activated again until the stimuli notices that there's product, and then the negative feedback kicks right back in. You can see another example of your feedback inhibition on the right-hand side of your diagram. So here we have the production of 3 different products, and what we see happening here is that, as you can notice, some of the boxes are blank. The blank are going to indicate our intermediate products because remember it's part of a cascading mechanism. And then we also notice the presence of lots of red lines. Notice how the red lines are coming off the products, lysine, methionine, isoleucine. Once those products are being produced, what we see happening is that the red line indicates that it's sending over an inhibitory signal so that we shut down the mechanics and we no longer produce any additional product because we have kind of reached our capacity or what we're looking for. So in a negative feedback system, you basically send out a stimuli. You turn on the machinery, and this would be your proteins or your enzymes. And as soon as you get to your final product, you send the signal back, removing the stimulus, and thereby shutting everything down. Okay? The negative feedback system, like I said, is the one that your body favors because it's basically a supply and demand type of relationship. You don't have to spend any extra energy. You don't produce any wasted product. You have exactly what you want. I do want to note, and this is not featured on the slide, that you also have a positive feedback system. In a positive feedback system, what we see happening is that you will send off the stimuli, you will turn on the machinery, and then when you produce your product, the product will also send a signal, but instead of the signal being an inhibitory one, it will actually be an amplified one. So as you produce the product sends a signal amplifying the stimulus and causing the machinery to continue to work. So as the end result, you produce even more product. The more product you produce, the stronger the signal becomes to keep the machinery on. This amplifying system tends to be very energy demanding, so your body will only utilize it in life-altering or life-saving mechanisms. So like for instance, platelets coagulation is an example of a positive feedback system. If you have damage to your blood vessels and you cause a cut in it to occur and now you're bleeding, your body will send a distress signal to allow platelets to accumulate at the site of injury. When one platelet appears at the site of injury, it's not enough to clot the opening. So the platelet will go ahead and send a stronger stimuli, thereby recruiting a second, a third, a fourth platelet, and it will keep amplifying the signal until it has hundreds, if not thousands, of platelets clumped together that can eventually block up the hole and stop the bleeding from occurring. All right, so negative feedback system means that the product will send the signal and shut down the machinery, whereas a positive feedback system will have more of an amplifying result, allowing the machinery to continue to work and obviously amplifying the amount of product that's being produced. Another way that proteins can be controlled is when we look at their overall formation of binding sites. We see that many enzymes can be labeled as allosteric enzymes. Allosteric means other shape, and it comes from the fact that when they were doing research to figure out exactly how enzymes can be turned on and off, they noticed that a lot of enzymes, if not most, have actually two binding sites. They have a binding site that's labeled their active site, which is where they can interact with their substrate. And then they have a secondary binding site that's going to be labeled a regulatory site. And on this regulatory site, they're able to interact with regulatory molecules, which will be a fantastic way to control when the enzyme is active and when it's inactive. So for instance, if you take a look at your illustration right over here, you're going to notice that on the enzyme that's been drawn, in order for the enzyme to be active, we can see that there's no molecule binding to the regulatory site, and then therefore the active site, it's maintaining its regular shape, and as a result, it's nice and on, and it's able to go ahead and interact with whatever ligand makes its way. On the other hand, what we see happening is when the enzyme is turned off, oftentimes there will be a regulatory protein that will bind over on the regulatory sites. And when it binds onto the regulatory site, it can cause a conformational change, among other things, to happen to the active site, thereby making it harder for additional ligands to bind. So by doing so, it will make the enzymes inactive, aka no longer being useful. It is only when the bound molecules are removed from the regulatory site that the active site then goes ahead and induces its original configuration, therefore increasing the likelihood that it can actively bind to the ligand and thereby continue its function. Not all regulatory proteins will actually cause a shutdown. There's actually also lots of examples where you can use a regulatory protein to actually induce what we call a positive regulation, meaning that you increase the effectiveness of the interaction between the ligand and the active site of the enzyme. So over here in this example, what we're seeing is that you have a regulatory protein, in this example, a regulatory molecule. In this case, we're looking at ADP, adeno-diphosphate molecule. And what we see happening is that it can be utilized to increase or change the conformation of the enzyme, thereby increasing its interaction with its ligand. So go ahead and take a look at your illustration, and as you can see, your enzyme has been featured in the green coloration. It has two parts with the dark green and the light green. Its substrate of choice is going to be a sugar. And what we're trying to do over here is we're going to go ahead and we're trying to oxidize the sugar so that we can extract energy out of it. And what we see happening is that the enzyme, when it's inactive, it has a completely different shape than when it's active. So notice how it's kind of like open versus closed, right? And what we see happening is that when it's in its closed conformation, that is when it's best able to interact with its substrate, the glucose molecule, featured in blue, and thereby we can basically increase the rate of oxidation so that we can get more ATP molecules, which we like to use for our chemical energy. In order to increase this effectiveness, we're going to be using a regulatory ligand, and in this case, that's going to be ADP. And as you notice, it almost looks like a little arrow format. It has its own binding site on the outside surface of the enzyme. And when it binds to it, it's almost like it's keeping the claw closed, for lack of a better description, which thereby increases the rate of catalyzation. So here we see illustration B. Notice how if we do not have our regulatory ligand, only about 10% of the glucose is able to interact with the active site on the enzyme. On the other hand, if we flood the system with the regulatory protein, what we see happening is a positive regulation. It's positive because it's going to increase the effectiveness of the enzyme, and it's doing so by inducing a conformational change. So once again, the claw, quote unquote, is going to close up, thereby allowing the glucose molecule to tightly bond to its active site, and as a result, we see that the enzyme becomes more effective. This is a little bit of a hypothetical scenario, but obviously what we see happening in our body is whenever our body becomes depleted of ATP, like maybe we are exercising, so we're using more ATP than normal, what we see happening is that the ATP molecule will dissociate into an ADP and a free-floating phosphate. So as our body notices that the ADP concentration goes up, it will actually also cause an increase in its positive regulation, allowing your body to seek out more sugar molecules so that you can oxidize it faster and thereby return or increase your ATP molecule production. Now, speaking of phosphates, phosphates are a very popular choice that we can use to control protein activity. And part of that is because a phosphate can be utilized, or the movement of the phosphate can help us control the formation of the ATP versus the ADP molecule. Now, I'm going to take a quick second because I want to make sure we all know what we're talking about. So ATP, adenosine triphosphate. The T is for tri. That means you have 3 phosphates within the molecule. And this is what we like to call the charged molecule. Oftentimes they'll make this little sunburst symbol around it. ATP is chemical energy. That's the energy that's generated courtesy of the powerhouse of the cell, aka the mitochondria. And what we see happening is when it releases its energy, it releases a phosphate molecule. So when it releases the energy, here's the energy. It does so by hydrolyzing its ATP molecule into ADP, adenosine diphosphate, which is 2 phosphate molecules, and a free-floating phosphate, okay? So we just basically separate the 3 in a batch of 2 and 1, and this now, the ADP molecule, this is what we call an uncharged molecule, meaning it doesn't carry any of our chemical energy. But it has the potential to link together with a phosphate, and when it does so, it creates an ATP molecule. So this is one of the ways that we can harbor our chemical energy. Now, when we start moving around that phosphate, we can go ahead and call it phosphorylation. Now, the phosphate will not always appear in an ATP molecule. It could also appear in a GTP molecule, which we'll talk about in a little bit. But in this case, what we see happening is, and we can take a look at the top of our illustration, is that the phosphate molecule itself will have 2 negative charges. And that will allow it, when it binds to the side chain of the amino acid of the protein, to cause a conformational change. And as it causes a conformational change, we can use that to either turn on or turn off the protein in question. So in our example right here, we can see that the ATP molecule will go ahead and remove the phosphate. It will phosphorylize, releasing the phosphate, which will then go over to the side chain of our protein. Notice how it says phosphorylated. And when it does so, what we see happening is that we change the shape, which means that we can either be turning it on or we could be turning it off. It all depends on what our ultimate goal is. Now, oftentimes when you do this, this is a very reversible process, and the phosphate, because it does increase the conformational changes of proteins, we can also utilize that to affect the overall binding of the ligand to the active site. Obviously, if we start changing the active site, the binding will not be as effective, making it less likely for the ligand to tightly bind, aka that the reaction will proceed. It will actually stop because of that relationship that can no longer be formed between the ligand and the active site. So the movement of a phosphate molecule is another way that we can control when the proteins are active or when they are in their inactive shape. In fact, what we see happening is that with phosphorylation itself, we can also go ahead and cause different covalent modifications to occur, and that can then be piggybacked to kind of control the actual location and interaction of secondary molecules within the protein itself. Now, over here, what we see happening is that our phosphate molecules are pictured here in yellow. When they start interacting with a protein, in this example, we're looking at a very well-studied protein. This is protein P53. P53 is what we call a regulatory protein. It's one of the ways that your body can handle DNA damage and stress being put on its genomic region. This protein can have control over the cell cycle. In essence, it can detect when a cell is mutated, and it can stop the cell from replicating, thereby inducing apoptosis. In fact, if you have mutated P53s, meaning that they don't function as they properly would, it actually increases your chances of developing cancer. So within our P53 model, as we can see right here, the phosphate molecules are going to alter or modify its appearance, because what's going to end up happening is they're going to create almost what we refer to as docking sites. And these docking sites are going to make it easier for other molecules to bind to our original protein. And as they're doing that, what we're doing is not only are we promoting the increase in size of the protein, so we're actually promoting the formation of larger molecules, but also what we see happening is that we can use it as a way for the protein to control when it's optimally being active versus inactive, and also responds to changing environmental entities, so thereby increasing or decreasing its overall rate of activation. Now, remember how I said to you that phosphorylation isn't only limited to the ATP molecule? Well, that's what we're seeing over here. So another way that we can control the protein is by utilizing GTP-Binding. GTP basically responds or correlates or refers, I'm sorry, I can't think of my words today, refers to guanosine's triphosphate. So once again, we have the 3 phosphate molecules that are on this GTP, and what ends up happening is that we're going to do hydrolysis, and when we do hydrolysis, we're going to remove a phosphate, and we're going to go from 3 to 2. So once again, we see that the active molecule is GTP and the inactive molecule is GDP. Now, some proteins are classified as GTP binding proteins. And what that means is that in order for them to be fully activated, they are going to need to have a GTP molecule bound to themselves. And when the GTP molecule binds, it turns on the enzyme or the protein, and it will continue to stay on until the GTP molecule hydrolyzes, meaning it uses water to pull itself apart. And remember, when it's pulling itself apart, that means it's releasing one of its phosphates, and we see that the GTP is then converted into GDP, and this particular one will go ahead and cause the enzyme to turn off and become inactive. and it will stay in its shut-off formation until the GTP molecule is dissociated. So notice right here on my chart, in order for me to go ahead and go from off to on, the first thing I'm going to require is for my GDP to dissociate, meaning that it needs to go away. This tends to be a rather slow process. And then what we see happening is the second thing that needs to happen is that we need to find another GTP molecule and get it to bind to the protein. And when it does that, we can go ahead and go back to our on/active formation. So keep in mind, in order for you to utilize GTP, we do have to have a GTP binding protein, meaning that there's an active site for it to accommodate the GTP molecule. Very similar to ATP to ADP, this process is completely reversible. As you can see, you just have to remove the inactive and bind back the GTP molecule to turn it on. And oftentimes, because the GTP is turning it on and the GDP is turning it off, we can go ahead and we can describe this relationship as acting as a molecular switch. Here you go. So the matching of a molecular switch, because one version is clearly on and the other version is clearly off. Another way that we can control proteins, and I really like this one, is the fact that we can go ahead and take proteins that are classified as motor proteins, and we can utilize them to move molecules across the cell. And we see this happening, for instance, along the scaffolding of the cell, when there are certain like hormones or different secretions that need to be brought outside the cell, or if there's a waste product that needs to be exported out of the cell. We also see it along muscle movements. So what we'll see happening is that you'll have your motor protein, and it will induce conformational changes that will allow the protein to kind of scoot down a scaffold. Now, the problem with this is that as the conformational changes are happening, it's really hard to kind of control it to make it move in a unidirectional way, meaning that you're allowing it to walk straight down the filament. So take a look at your picture right here, and what we're seeing happening is that our motor protein can basically take on three shapes: A, B, and C. Now, the three shapes are going to be important because it's going to allow us for some movement, but notice how by itself, we can't really control it moving in a straight pattern. In fact, what we see happening is that the motor protein is just kind of like wandering. It's just wobbling back and forth. So how do we solve this problem? What can we do to control the movement of this wobbling motor protein? Well, how about we introduce an ATP molecule and we use that chemical energy to almost propel it forward and make sure that we're able to control it from the starting point all the way to the end point. So let's go ahead and switch over and take a look at our next picture. And what we see happening here is that we have our motor protein in its three conformational shapes, A, B, and C. And what we're going to do is we're going to go ahead and introduce the step of binding an ATP molecule to it. And when the ATP molecule binds to it, as you can see, it will induce ATP hydrolysis, because we're going to use that energy from our ATP molecule to propel the motor protein forward. And as the ATP molecule hydrolyzes, remember it leaves behind ADP. Here's our little ADP. And there's our little phosphate that's exiting the system. And when this happens, this is pretty irreversible. Okay, there's no way for this motor protein to gather up the phosphate and the ADP and make it ATP again. Instead, what it will do is it's going to just go ahead and release the two components. It's going to release the phosphates and it's going to release the ADP. And as it does so, we see that as it changes back to its conformational change of A, it will await the introduction or the binding of the next ATP molecule, thereby once again shifting it forward. So this introduction of the ATP molecule makes it the way that the motor molecules can move in a one-way directional term, and they no longer wobble. So by repeating this process of ATP binding and hydrolyzing it, we're basically shoveling it forward so that it can move in a smooth, predictable way down the cytoskeleton filament. We also see that proteins will often work together, and when they do so, we like to call them machines. And protein machines are basically able to increase their complexity, and they're able to go ahead and create functions that are a lot more elaborate. So on our little illustration, what we're seeing over here is that we have several individual proteins that will have to work together to complete a task. And in order to be completely successful, oftentimes they will rely on hydrolyzing either ATP or GTP molecules, because this will allow them to drive the process. And as they're doing so, the movement of the phosphate itself will often induce some conformational changes, which makes it easier for them to turn on and off selective proteins within the complex or within the machinery. This then makes it able for them to turn, to have more of a coordinated movement between the different proteins. And obviously, we also want to have it somewhat be unidirectional, meaning that these proteins, they want to work together from start to finish. You don't want to have them go in a backwards flow. You want to keep them working until you get the product that's produced by their collaborative effort. The other thing that we should definitely talk about are these things called scaffolding proteins. So if you think about it, I've mentioned to you several times before that proteins need to interact with other molecules in order for them to fully function. Now, they can't just interact with any molecule, because one of the other things we've discussed is the fact that the binding site or the active site of proteins has high specificity. So it's kind of like our little lock and key analogy. So if you think about it, how would we go about explaining that the proper protein is matched with its proper ligand or substrate within the cytoplasm? We know that the cytoplasm is full of different molecules. So how do we make that connection? How do we get the ligand to match with its proper protein? Well, it turns out that we can use these structures called scaffold proteins. Scaffold proteins roam around the cytoplasm, and they can basically be described as very large molecules that contain multiple binding sites, which will allow them to interact with multiple proteins at the same time. And oftentimes what a scaffolding protein will do is it will collect sets of interacting proteins. So go ahead and take a look at your picture, and what we're seeing here in our first part is we have a scaffolding protein that has what we call different structure domains, which will allow it to interact with different proteins. So as you can see right here in green and in yellow and red, it's collected proteins or protein components that are compatible. And then it will also have these like unstructured regions. And the unstructured regions are going to be important because they're going to allow for flexibility to occur within the scaffolding protein. And these flexible regions are going to make it easy for the scaffolding proteins to increase the interaction between the proteins that are linked to their structured domain. So here you can see in my middle picture, you can see I'm using, or they're demonstrating that the elastic component or the flexibility component of the unstructured region is bringing the different interacting proteins closer together. And as you bring them closer together, you basically increase the probability that they're going to react to each other, thereby producing your ultimate complex that you're after. The real benefit of a scaffolding protein is not only does it increase your rate of chemical reaction because it's bringing together the compatible units or structure, but it can also be utilized to kind of isolate, or I should say confine the reaction within certain regions of the cell, thereby making it easier for the cell to identify where the reaction is occurring and where the product is going to be secreted from. One last item that I want to mention among how proteins are controlled is that it turns out that you can also have what they call weak interactions between the products or the macromolecules, and these can ultimately produce larger biochemical subcompartments. And a really good example of that is the nucleolus. So the nucleolus is an organelle that we have in eukaryotic cells. It sits within the nucleus. Its name of nucleolus literally means mini nucleus, and the nucleolus is the site of ribosome production. Now, how do we get a nucleolus? Well, it turns out that when we're doing our cell cycle and our chromosomes go back into interphase, certain chromosomes have genes that basically allow for the synthesis of a nucleolus, aka synthesis of ribosome production. So these chromosomes will each produce their individual nuclei. So those are the different products. And what we see happening is that these individual nuclei will start forming weak interactions with each other because they recognize their similarity. And if there's enough of them, they will actually fuse together and create one large subcompartment. And then this would be another way that we can go ahead and we can control the products that the proteins are being produced. So this right here is just a spherical liquid drop nuclei. And as you can see, the smaller parts are being converted into one larger entity. And this is then how we get our nucleolus, the production site of the ribosomes. I have some other examples on our next slide. So let's take a quick peek over there. All right, so here we can see our little protein scaffolding. We can see the products. Now, the accumulation of the protein products is also one of the ways that we get our amyloids. Remember, when the prions fold improperly, they create their little amyloid products. So it's not always a good thing, but you can also see that the intracellular condensation can be utilized to form our nucleolus. We can see it being used for our microtubules, and even in this example, we can see it being used to create a separate area right underneath the plasma membrane of this particular cell, right over here. So I like what it says over here. It says such intracellular condensates concentrate a set of macromolecules, thereby producing regions with a special biochemistry without the use of an encapsulating membrane. So unlike the rough ER and the smooth ER, they won't have a distinguishing membrane that will set them aside from the rest of the organelles in the cytoplasm. Now, our last portion of the chapter is going to take a look at exactly how we go about studying proteins. So what research methods are utilized to come up with the knowledge that we've just chit-chatted about in the 3 parts for chapter 4. And on this section, I'm just going to kind of skim over it. This is just going to be a little FYI because obviously we have so much detail that we've already discussed in the previous sites. Plus, a lot of these techniques we're going to come across once again as we continue to explore our chapters. So here's the breakdown. How are proteins studied? Well, the first thing that needs to be done is that the proteins need to be purified or separated from their sample, whether this is from a cell or a tissue sample. Then we need to go ahead and determine the protein structure by taking a look at how the amino acids are sequenced together, meaning what amino acids are composed or organized together to form this particular protein. We are also able to now take a look at large-scale production, design, and analysis of mostly any protein because of the fact that we can utilize different genetic engineering techniques. And last but not least, as is in many cases in biology, proteins will often, that are often seen having the same shape and structure will often be due to the fact that they are related to an ancestral protein, so they can be classified in what we like to call little protein families. So here is panel 4.3, and it just go ahead and just give it a nice little read through, but it basically talks about the fact that the first step in having to study proteins is the fact that we have to separate them from the cell and or the tissue that they are being housed in. So you can take a look at some of the experimental breakdowns, and it talks about the fact that you collect your sample, and then you basically have to go ahead and separate the organ or tissue that you're interested in, and from there on you're going to separate the cells and you're going to cause them to lyse. And then oftentimes we'll utilize differential centrifugation to spin the sample at different speeds. And when we do that, we see that the organelles, as well as the macromolecules and the proteins, anything we're interested in, will start separating in different layers or different segments, mostly based on quantity and overall size or mass. And when we do that, we're able to control which sections of the cell we're interested in. A centrifuge, for those of you who have heard the name and are trying to remember exactly what it does, it's a piece of lab equipment that can basically spin your sample at different RPMs. And as it's spinning it, it's often utilized to create a sediment at the bottom and then a liquid portion towards the top. Nowadays, you can easily buy kits that will help you with your protein purification and also isolation of your sample. So they really make it super easy for you. As long as you have your batch of cells, you go ahead and use the reagents within the kit, and it will cause the cells to lyse open, pop open, and then through the use of different filters and centrifugation, it will go ahead and isolate the proteins from the rest of the sample that you have within the cell and its extracellular material that it releases when it pops open. So here's just another look at protein separation. Another technique that you can use is you can do chromatography. Chromatography is a very popular choice when it comes to protein separation because it will allow you to go ahead and fraction your proteins. There are many different ways to do chromatography. You can do it through what they call an ion exchange, which is where you're basically using positive and negative charges. You can do a gel filtration, where you can basically control the size of the molecules by creating a different percentage gel, so making it harder for large molecules to move through it. You can also use an affinity chromatography. This is usually where you have a matrix that's covalently coupled to a molecule, so you can kind of control which protein you want to interact with. And as you're doing your chromatography, your end result is that you can kind of fraction or separate your sample into the specific proteins that you are after. Because as you now know, we have a lot of different diverse proteins within our cytoplasm. So it's definitely important to be able to separate them into the proteins that you're interested in studying. To continue looking at the separation techniques of proteins, oftentimes we can also use gel electrophoresis. It's the same old technique that some of you might have been exposed to in some of your previous labs, but you most likely use DNA, where you use the negative charge of the DNA to kind of run through a gel, an agarose gel, and in that case you can separate your DNA fragments with the larger pieces staying on top and the smaller pieces migrating towards the bottom. It's the same concept except for proteins naturally do not have a charge. So what you have to do is you have to use an SDS gel, which is a specialized type of detergent gel, and the SDS will actually interact with the protein, causing it to slightly unfold, and it will also allow it to get a negative charge. And because it now gets that negative charge, we can go ahead and introduce it to an electrical current, just like we would do with regular DNA electrophoresis, and then the protein fragments will start moving down the gel with the larger pieces staying towards the top and the smaller pieces migrating towards the bottom. So this can be another way that we can separate our protein. Once you have your overall protein that you're isolated from your sample, you can also want to go ahead and you want to take a look at exactly its amino acid sequences. So which of the 20 have been utilized and in what order have they been strung together. So a lot of times for that, we can use mass spectrometry. That's another technique that we can quickly figure out exactly what the peptide sequence is going to maintain. And here you can see some other ways that we can isolate the amino acid sequences. Crystallography is a popular choice. Spectroscopy is usually the one that we prefer. We can even use the electron microscope through its various techniques of beaming electrons at different frequencies, so that we can go ahead and we can figure out its three-dimensional shape, as well as which amino acids have been involved in the creation of the protein. And then last but not least, oftentimes as you study your proteins, you're going to notice that there are protein families. Protein families means that they often indicate very similar structure, as also very similar sequences of their amino acids, meaning that they all originated from what we like to call an ancestral protein. So this kind of comes into play, especially when you take a look at your larger proteins that are composed of multiple domains. We can then go ahead and separate them and see from which family they originated. I just want to point out that I do realize that for the section on how proteins are studied, I did rather swiftly move through it. And like I said, that's because we've covered a lot of information in this particular chapter. So for this last part, just kind of know in general what some of the techniques are. You really don't have to know it in great detail. You have lots of other stuff to kind of work on. Having said that, I think that this is where I'm going to end my lecture for Chapter 4. As always, if you have any questions or concerns, I love to talk to you during our Blackboard Collaborate sessions, but you don't have to wait. Feel free to send me that email or utilize your discussion sessions. All right, so we will talk soon. Bye.